Hello everyone. My name is Madeline Chung, and welcome to my presentation. This past month, I've had the absolute pleasure of conducting research under the guidance of my mentor, Dr. Brian Lin, and today I will present the information I learned about AlphaFold, a fascinating artificial intelligence system that has helped shape the field of science for years to come. And I'm so excited to share my findings with you, so let's begin. I've included my abstract on the left side of this slide for your reference. However, in summary, my research covered a lot of different aspects of AlphaFold, including AlphaFold's basic functions. I've also identified its main benefits and disadvantages, and I've also deep dived into some notable achievements made by researchers using AlphaFold already. And so, although it has contributed greatly to the science field, it's important to acknowledge its limitations. So while doing so, I explored some existing ways researchers have overcome these weaknesses. I've also proposed some unexplored opportunities for AlphaFold to be applied in order to advance the research progress, and suggested some potential improvements to be made in future updates of AlphaFold. And with the agenda laid out, let us dive into the world of AlphaFold. So what is AlphaFold? AlphaFold is a machine learning algorithm that functions using artificial intelligence to produce high-resolution structures or protein structure models. And the difference between AlphaFold and similar existing tools that have been existing for a long time already is that AlphaFold uses EvoFormer, which is a protein language model made up of 48 blocks that produces information about protein structures in a way that it is recycled over and over again. And to explain this in more simpler terms, this means that the information is not only produced as an output, but it re-enters the system numerous times as an input. And so every time it does this, continuous structural revision is made to the prediction, and each block does this over and over again until a very final accurate prediction is produced, which is similar to one shown on the screen, like coils. And with each prediction, AlphaFold will also generate some measures of accuracy. And this includes the confidence score, which is called the PLDDT score, as well as a prediction aligned error metric, the PAE metric. And so the confidence score estimates the probability of accuracy for each region of predicted structures, while the PAE value serves to measure the positional accuracy of some of these models. And so with that, now we understand how AlphaFold works. Let's talk about AlphaFold's very apparent strengths as an artificial intelligence tool. So first, AlphaFold has record-breaking accuracy. So at the 14th critical assessment for protein structure prediction challenge, which is a kind of test for different protein structure tools, AlphaFold consistently outperformed every competitor, scoring a 92.4 on the global distance test, which is a measure of protein structure prediction accuracy from 0 to 100. And higher values indicate a higher accuracy in those models. And so secondly, AlphaFold produces these predictions, highly accurate predictions, in a very timely manner. And it reduces the time-consuming nature that is typically associated with protein structure prediction. It takes only 1 GPU minute per 400 residue protein, compared to some similar tools like RosettaFold taking more than 10 minutes for the same task. And so RosettaFold is actually a similar tool to AlphaFold in a manner that it doesn't use AI, but it also produces protein structure predictions. And its lacking speed kind of emphasizes how AlphaFold needs to complete, or the need for AlphaFold to complete this task in a more efficient manner. And so alongside its timely abilities, AlphaFold also has an extensive coverage of information on protein structures. It covers a wide variety of protein structures, which includes information for 98.5% of the human proteome, which is important for other biotechnological scenarios I will discuss later. And with the introduction of AlphaFold, a study showed that proteins without structural information at all decreased from 5,027 to 29 because of the wide variety of information that AlphaFold can provide. And so now we've discussed kind of the strengths AlphaFold has. Let us look into some of the applications and how these strengths have been used by researchers to expedite processes and pioneer some new discoveries. So first, because of AlphaFold's access to 3D structures, we now have increased function predictions because we can kind of look at these high resolution models and comprehend these protein functions. So an example of this is because of AlphaFold's information, we have kind of accessed a structural explanation for the phosphatase and tensin homolog deleted on chromosome 10, also known as P10. And because of AlphaFold's 3D structures, we are now able to see the inner workings of P10 and understand its role of related proteins and detect some signal transduction pathways, which is really important to understanding P10's function and role in the human body. And similarly, due to AlphaFold, we have now expedited the process of drug discovery. Because of AlphaFold's access to 3D structures, we can also identify drug targets and easily select optimal ones. And because of that, we can analyze drug target networks, and completed analysis of this has been used in investigation of treatment against various conditions, including hypothermia and the monkeypox virus, which has been done by researchers already. AlphaFold has also been integrated into other databases in which its models is displayed alongside these other resources. And this includes databases such as the Swiss Model Repository, the European Protein Data Bank, as well as the String database. And interestingly enough, the String database is actually used for protein-protein interactions, which is not similar to AlphaFold whatsoever. However, it uses AlphaFold models to visually represent these interactions, which is very beneficial to researchers who are using these databases. And finally, AlphaFold is really beneficial for protein design because it provides information on disorder regions, which guides the experimental approach for manipulating these proteins for improved features. And these improved features include optimal functionality and like optimal stability or optimal size. And so while AlphaFold has made significant contributions through the notable achievements I just mentioned, it's important to mention its weaknesses and find some ways that researchers have overcame its limitations to use AlphaFold to its full potential. And so I will go over some of these. The first one being that it lacks information about some crucial aspects of protein structures. I've listed some on the screen, including multimeric structures and protein-protein interactions. And because of this lack of information, it becomes difficult to apply AlphaFold in a variety of situations. However, AlphaFold has been foundational in developing some solutions to this issue, including the AF2 complex, which is a separate system that uses AlphaFold information to actually predict protein-protein structures interactions. And similarly, the DeepMind team, which is the team responsible for making AlphaFold, has released an extension called AlphaFold Multimer, which provides information on multimeric structures, therefore covering for sort of this lack of information on these aspects. Similarly, AlphaFold has had some failures in drug development. And this is because while AlphaFold structures are very accurate, there is no consistency in the accuracy of amino acid side chains, which is crucial to drug discovery. And it also cannot detect ligand interactions because it lacks some of that key information. And because of that, it's really hard to kind of expedite the drug discovery process when there's some holes in knowledge when you are analyzing the drug target networks. And you might be wondering, well, previously, I already talked about AlphaFold being crucial in the process of drug discovery. And so the reason it has been possible to do this is because of this sort of solution of referring to other traditional methods and kind of experimentally solving these structures to find ligand interactions. And in fact, there is also a system that is called AlphaFill, and it uses AlphaFold predictions and fills them with information from experimentally successfully predicted structures that typically have more sequence identity than AlphaFold models. And so filling this knowledge gaps helps AlphaFold reach its full potential in the drug discovery process. Another limitation of AlphaFold is that it doesn't represent protein conformational diversity because it fails to kind of acknowledge those conformational changes. Studies have shown that AlphaFold only shows hollow forms of 67% of models, the others being apoforms. And so these two forms of protein structures are important because we want to access both. And because of AlphaFold, we can't really access the biologically relevant states that we need. And it is shown that the models actually lower in quality as the difference between apo and hollow forms increase. So another way to say this is that AlphaFold negatively correlates with kind of the conformational diversity there. And so it's really hard to accurately model the dynamic nature of proteins through AlphaFold. And some solutions that that researchers have proposed is actually trying to limit the input, the alignment depth of the input to only 16 sequences. And this does allow the system to account for some more conformational differences, but obviously this is a huge limitation in itself, is that we have to kind of manipulate the input there to produce the results that we want. And finally, the last limitation I have to discuss is mutations, because AlphaFold does not predict mutations in proteins in a variety of scenarios. And so wild-type structures and mutated structures show very little differences when displayed in AlphaFold, which is a very big problem considering that we need to detect these mutations in a variety of disease research. And so kind of segueing to my next section of disease research, AlphaFold has played a role in a lot of research studies, but I think I can propose some more unexplored uses of AlphaFold where it hasn't been applied, but I think it will be an effective tool to apply to the research process. And so I plan to suggest AlphaFold in relation to viral research, and specifically dengue viral research. And so dengue, just to provide a little bit of background, is the most rapidly spreading mosquito-borne viral illness and has the potential to be fatal. It causes 20,000 deaths a year. However, there's no medication that has passed clinical trials for the virus. And so I think AlphaFold has the potential to simplify the process of finding treatment for dengue in a very cost-efficient, timely manner, because artificial intelligence can be applied to the research process. So first, AlphaFold can predict the structures of dengue-related proteins, dengue being caused by the dengue virus. And so some of these structures it can provide are Claudin-1, which is a host cell surface protein that is essential for virus entry and the spread of dengue, or like lymphocyte antigen 6E. And so with the structures of some of these dengue-related proteins, we allow researchers to kind of gain a proficient understanding of the dengue virus on a more molecular level. And we are also able to test these proteins' tractability, which means its potential to be a drug target. And so with this information, researchers can easily observe drug target interactions, like I mentioned before, and test its interactions with specific medications that have shown potential anti-dengue effects, which includes some of the medications I've listed on the screen there. And so it's not enough for me to just tell you that AlphaFold can be potentially helpful in the dengue viral research field. And so I kind of wanted to provide you a visual representation of what I just mentioned. So here is an experimentally determined interaction between the dengue M protein and Claudin-1. And so we used AlphaFold multimer to produce these figures for my research paper. And I kind of just want to point out that on the right, kind of image B, there's some regions highlighted for the possibility of more in-depth functional studies, as they are called interaction sites, where AlphaFold can be applied here to in the manner I mentioned about drug discovery, and they're applicable to the experimental process. So that's kind of why I wanted to point them out. So here's kind of a figure I used in my research paper, and kind of a visual representation of what I was thinking or proposing for the future of AlphaFold research. And kind of related to the future of AlphaFold. Truly, the future of AlphaFold AI is very bright. Improvements and extensions are continuing to be made. The AI is continuing to be applied to a variety of biotechnological scenarios. However, since AlphaFold is ultimately a machine learning algorithm, it relies on pattern recognition to function, meaning it lacks some of the basic knowledge of protein physics, protein chemistry, and it can't predict some of these protein variants, which is kind of what we need in these more experimental scenarios. So I propose that we implement basic data on protein physics. And so now, with that information, the system can understand more environmental influences on proteins. And with that, we will be able to have access to conformational diversity and kind of address that limitation head on. And with that, we can account for these changes, account for these environmental influences like pH, temperature, ligand binding, and we can sort of access these dynamic nature of proteins, which we weren't able to access before. Similarly, we can implement some data on protein chemistry to understand the basic protein folding process and therefore addressing the limitation of not being able to see new mutations. And we can access some more biologically relevant states to the scenarios that AlphaFold is being used in. And so with that, now that we've kind of touched on AlphaFold's functions, its benefits and disadvantages, it's clear that AlphaFold has been a powerhouse in the field of structural biology, and it's produced so many high-resolution models with record-breaking accuracy, and especially in a timely manner with extensive coverage. And I think that despite its remarkable success compared to other tools, it's really difficult to ignore its weaknesses. And that kind of serves as a reminder that our artificial intelligence in general, and specifically this artificial intelligence system, can't really completely replace traditional experimental methods that researchers have been using for decades. And as researchers continue to combine other methods and other techniques with AlphaFold's robust capabilities, we can really embrace the endless possibilities and kind of expect to witness its continued success as it continues to accelerate progress in the science field. So with that, thank you so much for listening. I hope you have enjoyed my presentation, and this has truly been an incredible journey. So I want to thank my mentor, Dr. Brian Lin, from the University of British Columbia, for helping me through this process, guiding me, equipping me with the knowledge I needed to embark on this journey. And I've truly enjoyed my time here, gaining valuable experience in the research process. And thank you so much to the Talaria staff. So that's all from me.